When I was at school, I sometimes thought that studying without exams would be great fun. But exams can be fun too if you're assured of scoring very high marks. Well, we at Topper enable you to catch the tiger by its tail. Hi, I'm Arjun Bhagat and welcome to Exam Special. Today we're going to solve problems, share tips and lots more. So let's start with the first topic for today, biomolecules.
This unit of biomolecules carries a total of 4 marks in the 70-mark chemistry paper. In short, it carries 6% of the total marks. One can expect very short answer type questions worth a mark or short answer type questions worth 2 to 3 marks.
There were times when my mind would go blank after reading a question. That happened because of nervousness. Now, to avoid nervousness, let's do a short recap before we start attempting questions.
In this chapter, you must have learned carbohydrates and their classification into monosaccharides, oligosaccharides, and polysaccharides. We also learnt about the structure elucidation of glucose. You must have also studied about proteins, enzymes, vitamins, and nucleic acids.
You better make sure that you know all these topics by heart. I think now you'll be able to make a good strategy for your studies, but there's one more thing that you should know before attempting the questions, and that's what we'll tell you in our next segment, which is called Knowledge Dip. In this segment, we'll get to know the ability type on which you'll be tested on.
To give you a general overview, questions testing your knowledge and representative ability are asked from this chapter. You would often find the paper peppered with conceptual questions pertaining to the topic, proteins, vitamins, and carbohydrates as well.
Alright, it's time when we fasten our seatbelts. Now we'll start on our questions. First of all, it's important to understand the way you have to present those answers. For the same, we'll show you a model question. So all set to hit the bullseye with your knowledge? Let's take a look.
In this question, you have to write chemical equations for the reactions of glucose with acetic anhydride and ammoniacal silver nitrate solution. This question is a knowledge-based average question. Here we need to recall the reaction of glucose with the mentioned reactants. So it is advisable for students to remember all the reactions of glucose because any of them can be asked in the examination. Take a look at how such a question should be cracked. Acetylation of glucose with acetic anhydride gives glucose pentaacetate. And glucose reduces ammoniacal silver nitrate solution, that is, the Tollens reagent to metallic silver, and itself glucose is oxidized to gluconic acid. This question came in CBSE 2005 for 3 marks. Remember the correct structure of glucose, it's pentaacetate and gluconic acid.
Now that you've done a model question, I'm sure that you must be all geared up to do a few practice questions. It's always advisable to do warm-up exercises before the actual event with some practice questions. Here comes the first question of my favorite segment, Practice Makes Perfect.
Define the following terms with regards to proteins. 1. Peptide linkage. 2. Denaturation. This is a knowledge-based question which can be answered by applying the knowledge obtained while studying the chapter. Topper rates this 2-mark question as difficult. Peptide linkage is an amide linkage CONH form between a carboxylic acid COOH and amino group NH2 of two same or different amino acids, accompanied with the elimination of water molecule. And denaturation of proteins involves the disruption of the secondary and tertiary structure of protein by some strong physical or chemical means. In this case, the physical means may be the temperature change and the chemical means may be the reaction with the strong acid or base. Remember that denaturation of proteins pertains to the secondary and tertiary structure only.
At Topper, we believe in making even difficult questions easy. So now if a question like this one just discussed comes, I'm sure you'll be able to answer it without any difficulty.
What happens when D-glucose is treated with the following reagents? 1. Hydrogen iodide, 2. Bromine water, and 3. Nitric acid. This question is a knowledge-based question which requires you to recall the reaction of glucose with the specified reactants. We rate this 3-mark question as average. Remember, each part of this question is worth a mark, so answer accordingly. 1. On prolonged heating with HI, glucose forms N-hexane. 2. On reaction with a mild oxidizing agent like bromine water, glucose gets oxidized to 6-carbon carboxylic acid, gluconic acid. 3. Glucose gets oxidized and converted to saccharic acid on reaction with nitric acid, HNO3. Remember that glucose gets oxidized to gluconic acid in the presence of a mild oxidizing agent like bromine water, while in the presence of strong oxidizing agents like nitric acid, it gets oxidized to saccharic acid.
To ease your preparation process further, let's attempt another question.
A. Name the 3 major classes of carbohydrates and give an example of each of these classes. B. Answer the following. 1. What type of linkage is responsible for the primary structure of proteins? 2. Give an example of a globular protein. This is a knowledge-based average question which came in the year 2007 for 3 marks. It requires us to recall the topics classification of carbohydrates and proteins. Here goes the answer. A. The 3 major classes of carbohydrates are 1. Monosaccharides, examples of which are glucose and fructose. 2. Oligosaccharides, examples of which are maltose and sucrose. And 3. Polysaccharides, such as starch and glycogen. Coming to the second part of the question, peptide linkage is responsible for the primary structure of proteins. And the last part, an example of globular protein is insulin.
Well, we are halfway down. Loads more coming up, more practice questions and the HOTS question and tips to give your best shot at the exams as far as this topic is concerned. Let's now take a short break, or should I say, let me take a break as you solve a breaking question. Here goes.
What are the essential amino acids? Give one example. Answer this for a mark. This is a knowledge-based average question. We left you with a question before the break. Did you think about it? The question was, what are essential amino acids? Give one example. Take a look at the answer. Amino acids which cannot be synthesized in the body and must be obtained through diet are known as essential amino acids. Example, valine.
Welcome back to our exam special on biomolecules. Up till now, we've looked at a model question, 3 practice questions in which we covered the reactions of glucose with various reagents and some important terms related to proteins. But we aren't done yet. There are more practice questions left, so without wasting time, let's start off.
Here is the 4th one for you. Name the main disease caused due to the lack of the vitamin and the source for each of the following vitamins: B1, C, and D. This is a knowledge-based easy question. It requires us to recall the various kinds of vitamins, their sources, and the diseases caused by their deficiency. This question is a very scoring and less time-taking. It is advisable for students to learn and remember this topic very carefully because questions related to any of vitamins can be asked. As for the answer to this question, 1. Vitamin B1 is found in yeast, milk, green vegetables and cereals, and its deficiency causes beriberi, that is loss of appetite and retarded growth. 2. Vitamin C is found in citrus fruits, amla and green leafy vegetables. Its deficiency causes scurvy or bleeding gums. And 3. Vitamin D is found in egg yolk and fish. Its deficiency causes rickets or bone deformities in children and osteomalacia or soft bones and joint pain in adults. Don't confuse between the sources and deficiency diseases of various vitamins.
So now you know what you should have in your meals. Well, vitamins are really important for your growth. I'm not just talking about eating vitamins, but also studying them. Yes, it's a very important topic. So let's now take one more question from it.
Another question based on vitamins may ask you, what are the 2 types of vitamins depending upon their solubility? Explain giving examples. Why vitamin C cannot be stored in our body? This is a knowledge-based conceptual question which can be rated as average. It can come in your exams as a 3 marks question. To answer this, you should know that vitamins can be classified into 2 groups depending upon their solubility. That is, fat-soluble and water-soluble. Fat-soluble vitamins are those which are soluble in fat and oils, but insoluble in water. They are stored in liver and adipose tissue. For example, vitamin A, D, E, and K. The second type are water-soluble vitamins. As the name suggests, they are those vitamins which are soluble in water. Examples of water-soluble vitamins are B group vitamins and vitamin C. Now for the last part of the question, vitamin C cannot be stored in our body because it is soluble in water and is readily excreted in urine. Similarly, all other water-soluble vitamins cannot be stored in our body for the same reason. However, an important exception to this is vitamin B12. Remember the difference between fat-soluble vitamins and water-soluble vitamins. Also remember which vitamin falls under which category.
So was that enough of a vitamin dose? Well, jokes apart, it's a must for you to have a proper diet and get a good sleep. Take it from me, a healthy lifestyle is very important to excel in your exams. Let's now take our next question, which is conceptual in nature and is slightly tricky. Take a look.
How do you explain the amphoteric behavior of amino acids? This is a 2 marks question. Well, amphoteric behavior means showing both acidic and basic behavior. Amino acids contain both the acidic or carboxyl group and the basic or amino group in the same molecule. In aqueous solution, the carboxyl group can lose a proton and the amino group can accept a proton, giving rise to a dipolar ion known as Zwitter ion. So by forming a Zwitter ion, amino acids display amphoteric behavior. Remember that the Zwitter ion contains both positive and negative charges.
Now for the last practice question. This is a knowledge-based question and is rated as average. But remember, though it's placed last, it's not the least, as it gives you 3 marks if you get it right.
This question asks you, what are enzymes? And also lists 2 characteristics of enzymes. This question requires us to recall the topic, enzymes and their characteristics. Enzymes are biological catalysts responsible for catalyzing various chemical reactions occurring in living organisms. 2 characteristics of enzymes are: 1. They are globular proteins, and 2. They are very specific for a particular reaction and a particular substrate.
The practice question must have gotten you all warmed up. It's now time to go to our HOTS question. These questions have been introduced by the CBSE to test high-order thinking skills and probe students to think beyond the books. That's in sync with our ideology also. So take a look at the HOTS question that we have in store for you.
Write the important structural and functional differences between DNA and RNA. Read the question carefully and apply your knowledge of the structure of the DNA and RNA. Both these come under the topic nucleic acids. The logic behind the question is that both these nucleic acids differ in structure as well as function. So the answer must highlight these disparities specifically. Here goes the answer. Let's first look at the structural difference between the DNA and RNA. DNA contains 2-deoxyribose as sugar moiety, while RNA contains ribose sugar. The nitrogenous bases in DNA are adenine, guanine, cytosine, and thymine. While RNA also contains 4 bases, the first 3 bases are the same as in DNA, but the 4th one is uracil in place of thymine. And DNA is double-stranded, while RNA is single-stranded. Now for the functional difference between DNA and RNA. DNA is the chemical basis of heredity and is responsible for maintaining the identity of different species of organisms, while RNA is responsible for protein synthesis.
That was the HOTS question. I really hope that you're now confident to take up any challenge related to this topic. If you get stuck anywhere, feel free to reach out to our experts and get your queries answered within 24 hours by logging on to our website www.topperlearning.com. We are there for you. Now it's time for some exam tips without which no preparation is ever complete. So here they are.
The structure of biomolecules like nucleic acid, carbohydrate, and proteins should be studied and practiced on paper, because most of the questions are based on the structure. Remember that the classification of carbohydrates depends on the number of simpler units they give on hydrolysis. The classification of vitamins and their deficiency diseases should be remembered correctly. Also, the structural elucidation of glucose is a very important topic from exam point of view, because various questions can be asked in different ways, like the reaction of glucose with various reagents and the cyclic structure of glucose, etc. The study of DNA and RNA should be done in a comparative manner, because most of the questions asked involve differences like the difference between RNA and DNA and the difference between nucleotide and nucleoside. Remember the difference between primary, secondary and tertiary structure of proteins, since they greatly affect the various properties of proteins.
Apart from these tips, you must take a great deal of confidence and positive attitude to the exam hall. Think out of the box and work on your presentation skills. Apart from the regular preparation, of course, this will also help you a great deal. See you next time with another exam special. Take care and goodbye.